Okay, hi guys. This is about osmosis in plant cells compared to animal cells. So it's from National 5, Unit 1, Cell Biology, Key Area 2, Transport across cell membranes. If you're looking at this, you've probably been directed to this from one of the e-learning activities. And this is part 2 of Key Area 2, and this is the first video that I'm doing for it. Right. So the point of this mini little PowerPoint is to look at the differences between animal and plant cells and how they behave in changing water concentrations. So something happens to an animal cell when you put it into pure water, and it's actually different from what happens to a plant cell if you put it into pure water. And there's a few new terms we'll come across. Two you've probably not heard before are turgid and plasmolysed. And the point of this is to just get familiar with both of them. Now, before we go into details of how plant cells and animal cells will behave differently when water is entering them or exiting them, we have to think about cell structure. What structure do plant cells have that animal cells don't have that might cause a difference in how they react when water comes into them? Now, we're going to come back to that question, but have a little think now. You should already know about 3 structures that plant cells have that animal cells don't. Which one might influence how they respond to osmosis, water coming in or water going out? So we're going to look at plant cells first of all. What we have, I'm just going to get myself a wee pen. What we have here is a normal plant cell. Okay, it's got a vacuole, it's got a cell membrane, and it's got a cell wall. Now remember, the cell wall is freely permeable, meaning that things can go across it, in and out, as much as they like. It's the cell membrane that's selectively permeable. Only some stuff can go across the cell membrane. Okay. Now, if we put that normal plant cell into pure water, so it's got high water concentration, it's going to become something called turgid. Now, what I need you to see is that the vacuole is much bigger in the turgid cell, and it actually ends up pushing out, and the cell wall of the plant cell starts to bend outwards, and you can see the cells actually increased in size. If we take our normal plant cell and put it into a solution that's got equal concentrations of water as to the inside of the cell, so that what we're saying is whatever percentage of salt and sugar is inside the cell, it's the same outside the cell. So there's no concentration gradient, okay? There'll be water moving in and water moving out at the same rate. There's going to be no change in the cell size, which means we just say that there is no change, okay? If we were to put that normal plant cell into a low water concentration, so maybe like a really salty solution or a solution with lots of sugar in it, meaning that there is more water inside the cell than outside, so the water, by osmosis, moves along the concentration gradient, will move out of the cell to the solution. Now if you look here, it becomes something called plasmolysed. And what I need you to notice is the vacuole is very small because there's no water in it anymore. And in actual fact, the cell membrane is starting to come in off the sides. Okay, starting to come in, so the cell will just shrink just a little bit. But what's really important, with a turgid cell, a plasmolysed cell, and a cell where there's no change, it's still a rectangular shape because that cell wall gives it a really firm structure. Here we've just got 2 little diagrams summarizing that. So if the water goes into the plant cell, which would mean if it was in like a pure water, so there's more water outside, the cell is becoming turgid. Okay, the vacuole is getting bigger, everything's pushing out against the cell wall. If your plant cell gets put into a really salty solution or sugary solution, so that means that there's more water inside the cell, and then the water is going to go from the high concentration inside the cell to outside. The water will move out. We get our plasmolysed cell when the vacuole shrinks and the cell membrane comes away from the cell wall. So the cell can go whoop in a little bit. But remember, they're both still rectangular. But if we look at our animal cell, here we've got our normal animal cell, and no cell wall, no vacuole, it's just got the cell membrane. If we put that into pure water, so we're going to have a high concentration of water outside the cell and a low concentration of water inside the cell. So the osmosis means that the water is going to move into the cell. We actually get our cell bursting here. Okay, there's no cell wall to stop it from bursting, so the cytoplasm takes in so much water that it just bursts out the cell membrane and the cell's basically destroyed. Now, if we take our normal cell and keep it in a solution that's got the same water concentration as what's inside the cell, there's no change. The amount of water going in and out is equal, so there's no change in the cell. If we put our normal animal cell into a low water concentration, so maybe it's a sugar solution or a salt solution, you see here that the cell shrinks. Okay, the cell membrane just comes in on itself because there's all the water is leaving it, so the cytoplasm is taking up less space. So we're getting a shrinking cell. Big thing here is, remember, animal cells they'll change their shape. There's nothing keeping their cell nice and structured. There's no cell wall. So that means that it can burst or it can shrink. Okay. So here we've just got our red blood cell as our example cell with that example. So you've got the water going in here. So that's when it's in pure water. The animal cell is going to burst because the low water concentration is inside the cell. The water will move into the red blood cell because remember, the water is going from a high concentration to a low concentration over a selectively permeable membrane. That's osmosis. Our middle one, we've got our normal one, and you can see here that there's water going in and going out, so it's at equal measure, so there's nothing changes there. Now, if we put our cell into our really salty solution or sugary solution, we're going to have a lower water concentration out of the cell, so because water is moving from a high concentration inside the cell to low concentration outside the cell, the water moves out, and our cell either shrinks or shrivels. Okay. So this is going to be a little summary comparing our plant and animal cells. Now, if the water is moving out, so if the cell is in a salty solution, somewhere with low water concentration compared to the inside of the cell, your plant cell becomes plasmolysed and your animal cell becomes shriveled or it shrinks. If we've got a situation where water is moving in and out of the cell at equal rates, no change. Both cells are normal. Okay. If we've got a situation where water is moving in, so the cell has been placed somewhere with a high concentration of water, meaning that there's more water outside than there is inside, the water is going to move in and our plant cell will become turgid and our animal cell will burst. Now, it's at this point, if you're in class, I'd normally stop this and ask you to take a wee note. So you might want to pause that and do that now. Right, we're just going to turn to our question at the start. What structure do plant cells have that animal cells don't that might cause a difference in how they respond to water entering or exiting the cell? Hopefully you've all got it clear in your heads now. It is, of course, the cell wall. Okay, it's really rigid and structured and it keeps that nice rectangular shape and it's very difficult to break. Animal cells don't have it. The cell membrane is weak, so when the water moves in, can't hold it, just bursts. Okay, and if the water moves out, it's going to change its shape as it shrivels up. This should also be a note that I'd have you be taking in class. That summarises that wee little section. What we've done is we've explained why animal and plant cells behave differently. It's all about the cell wall. Okay, and we've got our new terms. Turgid is a plant cell when water has entered it, and plasmolysed is when the plant cell, when the water leaves the plant cell. Now, animals, they're either going to burst when the water goes in, or they're going to shrivel or shrink when the water comes out.